Hello students, welcome to the class. Today we will be discussing on an important you can say it can be a 10-mark question in the exam, or it can be a 15-mark question, or sometimes it can be a 3-mark or 2-mark, very short answer question. Okay, short essay, long essay, or short answer question. It can be anything out of all this. In the today's session, we will be discussing what are isoenzymes. Then we'll also be discussing the different types of isoenzymes, and also we are going to discuss some examples of the isoenzymes. So these are the three things what we are going to see in today's session. So coming to isoenzymes, we should always know a few things, which includes why they are called isoenzymes. Okay, we normally know iso term is used whenever there is same. Okay, they are same. And isoenzyme means they are similar enzymes or same enzymes. In terms of what? Similar enzymes. In terms of their reaction, means type of reaction or type of substrate to product conversion, it is same. So they are called as isoenzymes. So otherwise, it is also called as like multiform of same enzyme. Like for example, A is converted to B. Okay, the reaction is A conversion to B. There are enzymes which are synthesized from different places or tissues. Like say about enzyme E1. The same enzyme is synthesized from say about muscles. It is synthesized from brain tissue, or it is synthesized from say about intestine. They all are different enzymes synthesized from different cells by the different genome system or gene. But what is common in them is reaction, type of reaction they are taking part of. They are different enzymes, but taking part in single type of reaction, that is conversion of A to B. So they are called as isoenzymes, or also called as multiform of same enzyme, because they are synthesized from different cells. If it is synthesized from same cell, they are the same enzyme. There is no change because same gene will dictate the synthesis of that protein. Here, it is synthesized from 3 different tissues. A particular enzyme, say about E1 is the one enzyme. You have E1A, E1B, and E1C. It is reacting same A to B, but they are different. The enzyme is completely different from each other. Only common is they are reacting the same reaction. They are called as isoenzymes, or also called as multiform of same enzyme. They are multiform of same enzyme synthesized from different tissue. Okay, they are different tissue, but taking part in same kind of reaction, same reactions. Okay, they take part in same reactions. Like A to B conversion is the reaction, A is the substrate, B is the product, enzyme E1 is the major enzyme. So it is like look alike. They all are look alike, performing same function. But they are born out of different mothers. Okay, they are friends born out of different mothers. So these are called as isoenzymes. Iso means same type of reaction they are helping out. The enzymes are taking part in same type of reaction. They are called as isoenzymes, or sometimes also called as isozymes. Okay, isozymes are also called as isoenzymes. So isoenzymes, if I tell you again, it is a multiform of same enzyme catalyzing the same reaction. They are called as isozymes or isoenzymes. So this is about intro of isoenzymes. What are isoenzymes? Now, when we see characteristics, okay, when we see the characteristics, isoenzymes.
Sir?
Yes.
Sir, as they produced by different genes, so the amino acid sequence will be different.
I could not hear you. Yes.
Sir, as they are produced from like different genes, so their amino acid sequence will be different.
Yes, their formation itself will be different. The formation itself will be different because they are synthesized from different gene only. They are different gene which are part of different cell. They are completely a different individuals. But what is common in them, isoenzyme, is they are acting on same type of reaction. Okay, they are called as isoenzymes. Now, if I tell you, their only common is reaction. Okay, they are taking part in same type of reaction. What is different is their physical nature, physical characteristics. Okay, their chemical composition, as I told, the amino acid sequence or amino acid numbers, or even the protein structure itself. it can be different. Okay. Or even if you take electrophoresis, that is their charge, electrophoretic pattern. Okay. Electrophoresis can be different. Or if you do chromatography, chromatographic pattern is also different. Or if you see immunogenic reactivity, immunological, they are different. Why? Because they are formed from different cells. They are formed from different cells, but they take part in same type of reaction. They are called as isoenzymes. So they are only common is reaction type. Otherwise, they are physically different, chemically different, electrophoretically different, chromatographic pattern is different, and also their immunogenicity, how much they act as a immunogenic stimulant, how much they will act as an antigen. That also will be differing from different to different. Not only these, they might also vary in, okay, they also vary in Km value, that is affinity towards the substrate. Maximum velocity of each isoenzyme can be different, or optimum pH. What we call optimum pH is pH at which the activity of an enzyme is highest. Okay, that is the optimum pH. They can be different in optimum pH also, or they can be differ in inhibitor affinity or influenced by the inhibitor, that is susceptibility. Okay, how much they are susceptible for the inhibitors. So they not only these characteristics are different, but also they may vary in Km value, Vmax value, or their optimum pH, or how much they are inhibited, susceptibility towards inhibitor, sometimes even denaturation. Okay, they differ in the denaturation also. That is degree of denaturation or degree of denaturation. So the isoenzymes are physically, chemically, electrophoretically, chromatographically, and immunologically different from each other, but they are common with reaction type. Apart from these characteristics, they also differ in their constant, that is Km, maximum velocity, optimum pH, susceptibility towards inhibitors, and also degree of denaturation of the protein. So this is a isoenzyme, the characteristics of isoenzyme. Now, as I told you, why they differ in all these characteristics is because they are formed from different gene itself, different genome, because they are formed in different cells from a different gene coding for the particular enzyme. So that is about the isoenzymes. Okay, that is the definition and characteristics of the isoenzyme. Next, in exam, they'll ask you define isoenzyme, name some examples, and describe any one of the isoenzyme. So when we see the examples, we come across four most common, and that three most common. One, that is the LDH, that is lactate dehydrogenase. Okay, in 3 marks question also, there is short answer. They might ask you give examples for isoenzymes. So you should be able to write the examples for the isoenzymes. First, LDH, lactate dehydrogenase. Also, we have got CPK or CK. Okay, that is creatine phosphokinase. Okay, or also called as creatine kinase. So that's why either we call it a CK or CPK, creatine phosphokinase. So this is the second example. Third is ALP, that is alkaline phosphatase enzyme. And fourth, very less told, that is alcohol dehydrogenase. Okay, so these are the 4 examples for the isoenzymes. What are they? They are reacting the same type of reaction, but they are different in their all the characteristics, all the physical and chemical characteristics by nature. So this is about the examples of isoenzymes. That is LDH, lactate dehydrogenase, CK or CPK, which is the creatine phosphokinase or creatine kinase, ALP, which is alkaline phosphatase, and alcohol dehydrogenase. So these are the 4. If they ask you 3 mark, define isoenzyme and write example of isoenzyme. You can define the isoenzyme and then write any 3 of these examples. Or sometimes they'll ask you just to write about the examples for the isoenzymes. So you can write all 4 examples. Or sometimes they'll ask you individually, like write isoenzymes of LDH. It can be a 3 mark question. Write isoenzyme of CK or CPK. Write isoenzymes of alkaline phosphatase. So these can be individually also 3 mark question. Okay, they'll ask you write the examples of isoenzymes. What are the types of isoenzymes? This is only the examples. Now we'll be seeing individually each. Okay, because each can be again 5 mark or 3 mark question. Okay, how we are going to classify these isoenzymes, which tissue is synthesizing these isoenzymes, and how they are significant. Because if we are learning isoenzymes, there should be some significance of it. There should be some utility of that isoenzyme. Otherwise, there is no point in understanding the different isoenzymes. Okay, so there is biochemical or clinical significance of each isoenzyme? So that can be a important question in the exam. So we'll be seeing in further slides. Till here we are clear with the examples?
Yes, sir.
Yes, very good. So we'll moving ahead. First, we'll see about the lactate dehydrogenase. Lactate dehydrogenase. Have you come across lactate dehydrogenase? Yes, it helps in catalyzing the interconversion of one important compound of glycolysis pathway, that is pyruvate, to lactate conversion is by the lactate dehydrogenase enzyme. So this enzyme is taking part in interconversion of a major intermediate of the glycolysis pathway, which is the pyruvate. So pyruvate conversion to lactate or lactate conversion to pyruvate, lactate dehydrogenase will take part in this reaction. Okay, that is about LDH. Now, what are the types of LDH? There are total 5 distinct isomers. There are 5 distinct isomers or 5 distinct isoenzymes. Means what? The LDH has 5 different types of brothers. Okay, there are 5 brothers from different cells for LDH. We number them. Okay, what are it and how we are going to number, we'll see. But how we are going to, how it was separated was when the LDH was allowed to run on electrophoresis. Okay, when it was allowed to run on electrophoresis, we all know the charged molecules move from positive to negative. Okay, we know they move in one direction. Okay, normally we see the direction from negative to positive node. So when it was added, okay, when the sample is added, they start migrating. They start migrating along with the electrons. In the migration, it depends on 2 things. One is mass, how bulky the molecule is. Second is how much they carry the charge, that is the negative charge. How much they are either positive or negative charge. Depending on that, it will form a ratio of mass to charge ratio. Okay, mass to charge ratio, which will define in particular time, like say about for 1 minute, you allow this circuit to run. In 1 minute, how much protein is running on the electrophoresis? That will depend on its mass to charge ratio. When they have separated that on electrophoresis, they could see that there are 5 points. Okay, 5 places where LDH has stopped running. So depending on that, they identified that LDH has got 5 different isoenzymes or 5 different isomers. And this LDH is a what do you call the enzyme which contains 4 subunits. How many? 4 subunits. Like hemoglobin contains 4 subunits, 2 alpha, 2 beta. Similarly, LDH has got 4 different subunits, which we call from alphabets H and M combination. Okay, H and M combination. So there is a combination of making 4. It can be all 4 H, 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 or it can be replacing H with the M. Okay, like for example, it can be HHM or H, H, M, M, H, M 3 times, or all are 4 M. So it can make combination like this. How many subunits are there? 4 subunits. 4 subunits make a combination to form 5 different types of isoenzymes. It can start with all H to all M subunits, same type of subunits. Based on this, we have got LDH of 5 isoenzymes, 5 types, 5 isoenzymes of LDH, which we have actually identified based on the electrophoretic mobility. Electrophoretic mobility. When we see the electrophoretic mobility, LDH, okay, LDH 1 is fastest moving. First, we'll see what are the different types. So we have numbered them as LDH 1, 2, 3, 4, and 5. Okay, LDH, LDH, and LDH. So these are the 5 different types of isotypes of LDH. 1 to 5. We numbered from 1 to 5 based on electrophoretic pattern. The one which is moving faster is the one, LDH 1, and least or less mobility which is showing is the LDH 5. And in between we have 2, 3, and 4, LDH 4. So from negative to positive node on electrophoresis, we identified it's the same protein or same enzyme, actually same enzyme acting on same type of reaction. When we allow them to separate on electrophoresis, we identified that this one molecule, one type of reacting enzyme is made up of different types of enzymes, or they are different on their electrophoretic mobility. So based on that, we have identified LDH 1 to LDH 5. Okay, so this is about the types. Now, what are they composed of? How they are composed of? So this is an important thing to be written in the exam, because we should know when we are telling there are 5 isoenzymes. Okay, when we are telling it's a 5 isoenzymes, we are also making a statement that it is synthesized from different cells or different tissue. Like for example, if it is synthesized by heart muscles, when there is damage to the heart, then there is particular set of isoenzyme which will be elevated very high. So that's why it is most important to understand why or which tissue it is synthesized from. So what are they? We'll see. LDH1, LDH2, LDH3, LDH4, and last one is LDH5. 5 different types of isoenzymes. We'll see what are the subunits it is made up of. Subunit it is made up of is first is always only H. All 4 are H. Okay, so all 4 are H. That is H subunit of protein. LDH1. LDH2, now what we will do is it is replaced with one M subunit. LDH3, 2H, 2M. LDH4, 1H, remaining all are M subunit. And last one, LDH5 is completely made up of 4 M subunits. So it is made up of 2 types of subunits, 2 types, like hemoglobin, 2 alpha, 2 beta. It contains 1 alpha subunit and beta subunit. Same way, LDH contains H and M. It is combination of these making 4 tetramer. Okay, 1, 2, 3, 4. So first one, LDH1 is purely H dominant. All 4 subunits are mono, same H subunit. LDH5 is all 4 are M subunit. Between one H is replaced with one M. This is about subunits. Next, we should know origin, from where they are originated, origin of that particular isoenzyme. Now, LDH1 is mainly from the heart tissue and RBC cells. Okay, it is mainly synthesized from the heart and RBC cells. Means these are the only 2 tissue which will synthesize LDH1. Similarly, LDH2 also, it is from heart and RBC cells. So they are synthesized from heart and RBC cell. What? LDH1 and LDH2. LDH3, it is mainly from brain and kidney. This type of isoenzyme, that is LDH3, is mainly from brain tissue and renal tissue, kidney, will synthesize LDH3 type. LDH4 is from the liver and skeletal muscles. That is the skeletal muscles, and liver will synthesize LDH4, this type of isoenzyme. And last one, isoenzyme LDH5 is mainly from skeletal muscle and little from liver. Last one, M stands for, you can remember, muscle. H stands for heart. Okay, the H predominant is mainly the heart. Then once the M predominates, M is skeletal muscle. Little above, that is LDH4, liver and muscle. When it is 50-50%, H subunit and M subunit are 50-50%, it is mainly from the brain and the renal system. So that is how you can remember. All 4H, heart and RBC. Second LDH also same, heart and RBC. When 2H are there, 2M are there, it is mainly from the brain and renal tissue or kidney. The next remaining is liver and muscle. And last LDH5, M stands for muscle. So mainly from skeletal muscle, little from the liver. This is about origin, from which tissue it is originated. Okay. And what do you see on electrophoretic pattern? Electrophoresis, I told you LDH1 runs very fast. So it is the fastest. Next, all are at the lower side. Next will be faster. This is just fast, slow, and slowest. So when you see on electrophoretic pattern from negative to positive node, how much it is fastly it is moving towards the anode or positive node. If it is running very fast, fastest, LDH runs very fast. Next, second position taken by LDH2, third position by LDH3, fourth by 4, and fifth by LDH5. That's why they have numbered as 1, 2, 3, 4, 5. And when they seen which tissue is synthesizing this kind of isoenzyme, one is from the heart and RBC cells. Okay, then LDH2 is heart and RBC cell, brain tissue, liver tissue, and significantly by the muscles. Okay, so this is about the LDH and how much they are making the plasma LDH. When you see total LDH, the total concentration of LDH, in that what is the proportion? The major proportion is the LDH2, 35%. Second is the 25% here, and also LDH3, 27%. Remaining LDH 4 and 5, they make somewhere about 8% and 5%. So less concentration is LDH5, little less is LDH4. Majorly, these three makes the major concentration of the total 100% of the LDH which is present in the blood. When you see total LDH, it is made up of 5 different subfractions. In that LDH1 to LDH5. So in that, it is higher concentration is LDH1, 2 and 3. So they are the 3. Why we have to know? Normally, we have seen LDH2 is higher when compared to LDH1. Okay, we have seen 35 and 27, 35%. It is 25%. In certain conditions like myocardial infarction, the LDH1 concentration increases. It increases very high because the tissue which is synthesizing, okay, the cardiac system which is synthesizing is damaged. When there is damage, always there is leak of the enzymes or the proteins or the substance into the blood. So when it is leaked into the blood, definitely the concentration of LDH is elevated. When you see which type of LDH is elevated, it is the LDH1, which is elevated in the myocardial infarction. So normal pattern is what? LDH2 is more than LDH1. It is normal. When there is myocardial infarction, LDH1 is more than LDH2, which we see in MI, which we call as reversal of the pattern. Okay, there is reversal of concentration of LDH1 and 2. Now we also know LDH, say about LDH4 and 5. In liver diseases, instead of 8 and 5%, its concentration increases. So in liver disease, it is elevated. So that is about the LDH or lactate dehydrogenase. Similarly, we have seen RBC cell also contain lot of LDH. Say about if there is blood sample you have collected and the blood sample is hemolyzed. Okay, the blood sample is hemolyzed. Now what happens? The concentration of LDH will increase in the blood, which is actually not because of any disease, just because the cells have ruptured inside the sample, the tube. So that is called as false elevation. False elevation in the LDH levels. So that is about the lactate dehydrogenase. Okay. Now second, we'll be seeing about creatine kinase or CPK, creatine phosphokinase. This helps in conversion of one important reaction, that is interconversion of or creatine phosphate to creatine. Okay, creatine phosphate to creatine. We have seen this reaction in the glycine metabolism, where we are synthesizing the creatine and end result is creatinine. Okay, so interconversion of creatine phosphate to creatine and creatine to creatine phosphate is regulated by CK. Now this CK has got 3 isoenzymes. How many isoenzymes is? 3 isoenzymes. So 3 isoenzymes takes part in the reaction. And again, how they are separated based on electrophoretic pattern. We have CK1, CK2, and CK3. Creatine kinase 1, creatine kinase 2, and creatine kinase 3. What are the subunits? Subunits we have either B or M. Okay, it is made up of B and M. CK1, it starts with B. Alphabet B is first always. BB. CK2, 1B is replaced by M, or also called as MB. And CK3 is purely by the M. What is the origin? Please remember, B stands for brain and M stands for muscle. So CK1 is synthesized by the brain tissue. CK3 is purely dominant by MM. It is by the muscle, skeletal muscle. In between, what is left is the hard tissue. CKMB. Okay, that is also called as CKBB, CKMB, and CKMM. So 3 isoforms or isoenzymes CK1, CK2, CK3 are also called as CKBB, MB, and MM. BB, B stands for brain, synthesized by brain tissue. MM stands for muscle, synthesized by muscle tissue. And last, CKMB, synthesized by cardiac tissue or heart tissue. So normally, what is the significance of this? Like for example, you were riding bicycle and you fell down and you had some muscle injury. When you test for creatine kinase concentration in blood, total is elevated. When you do isoenzymes, which type it is elevated? It will be the CKMM because it is present high concentration in muscle. When muscle is damaged, the concentration of this amount or the enzymes will be excreted in or escaped into the blood tissue. So that time you will see high concentration of CKMM. Like for example, the person has got MI, myocardial infarction. What type of isoenzyme is elevated is the one which is originated from the cardiac system. That is the CKMB. That's why we have one test called as CKMB analysis. Okay, that is creatine kinase specific isoenzyme to the cardiac tissue. If person you are identifying is having cardiac arrest or myocardial infarction, like you do the troponins, you can also do specific isoenzyme of CK, that is creatine kinase MB, because it is majorly synthesized from the cardiac system. Okay, so that is the importance of this CK. That is the significance of creatine kinase. How many isoenzymes? 3 isoenzymes. How many subunits? It has got 2 subunits or dimers. One is B, other is M. BB is first form CK1, MB is CK2, and CK3 is MM. BB stands for brain, synthesized by the brain tissue. MB stands for the heart, cardiac system. MM stands for only muscles. This isoenzyme is elevated in the muscle damage. So that is about the CK or creatine kinase. I hope you are following. Okay. Yes. Now third one, another most important is ALP, that is alkaline phosphatase. Okay, alkaline phosphatase. It has got around 6 isoenzymes. Okay, it has got around 6 isoenzymes, and ALP is a monomer. There are no combinations. It's a monomeric enzyme. It's a monomer enzyme, single enzyme. But what is different in each isoenzyme is the concentration of carbohydrate in the protein. Okay, amount of carbohydrate in the protein is different in different isoenzymes. 6 isoenzymes differ only in the percentage of concentration. Okay, now we'll see what are the 6 different isoenzymes. This itself can be a 5-mark question or 3-mark question, isoenzymes of the ALP. So first one, that is alpha alkaline phosphatase. So alpha alkaline phosphatase, it is synthesized from biliary canaliculi. Okay, when you see the gallbladder, the major ALP which is synthesized by the gallbladder, okay, biliary epithelial or canaliculi, it is synthesizing epithelial cells will synthesize this type of isoenzyme, ALP, alpha ALP. It constitutes somewhere around 10% of total ALP. Like say about it was 100% of ALP, in that 10% is contributed by the gallbladder or biliary canaliculi, that is the epithelial cell. When you see elevation, like for example, you have got obstructive kind of jaundice where stone is formed in the gallbladder. Now, when gallbladder is eroded by this stone, this ALP will elevated. Okay, in obstructive jaundice, this type, that is alpha ALP, is elevated in the blood. So that is about the first one. Second one is alpha 2 heat labile. Heat labile means they are unstable for the heat. If you give the heat, they'll be destroyed. They are stable up to 56 degrees Celsius. If you increase the temperature, say about 65 degrees Celsius, the protein get inactivated. The enzyme get inactivated. They are heat level, not they are sensitive to the heat. Majorly, they are produced by liver cells or hepatic cells. How much is the concentration? Major portion, 25% of total. So whenever there is liver damage or hepatitis or some kind of infection to the liver, the type of ALP which is elevated is the alpha 2 heat labile. Alpha 2 heat labile type of isoenzymes. Okay, this is about B. Now C, there is type of alpha 2 heat stable. Means if you give this temperature, that is 65 degrees Celsius, still enzyme is active. Okay, still the enzyme is active and they are functional. So this is majorly seen in the what you call similar to the placental isoenzyme. Okay, placental isoenzyme. So this is 3 what we have seen. Next, D, that is also called as pre-beta ALP. Pre-beta ALP. Now this type, it originates from bones. Okay, the ALP, alkaline phosphatase, because in formation of bones, you might have read about 2 enzymes which play a major role in mineralization and demineralization. That is acid phosphatase and alkaline phosphatase. It will create an environment in the bone tissue where it can become acidic environment or basic environment, alkaline environment. Alkaline environment given by alkaline phosphatase, Acidic environment is given by acid phosphatase. So bone tissue synthesis ALP, what type it is pre-beta. So if there is bone disease, okay, if there is bone disease, the pre-beta type will be elevated. Okay, pre-beta type will be elevated. What is the normal concentration of total? 50% of the total ALP is pre-beta type, means major portion which is present in the blood is majorly from this origin, bone origin. Next, E type, that is the gamma ALP. Okay, why they are called alpha, gamma, beta, I'll tell you next. Okay, next one is gamma ALP, which is originated from intestinal tissue or intestinal cells. So whenever there is some disorders of intestine, like say about ulcerative colitis. Okay, say about ulcerative colitis, because it is a disease of intestine. The intestine contains lot of this kind of ALP. When there is damage to the epithelial cells of intestine, it gets leaked into the blood, and the concentration of this isoenzyme will get elevated. That is elevated. Normally, it is only 10% of total ALP. Whenever there is damage, it will increase more than 10%. Now, last type, there is ALP present in leukocytes. Okay, leukocytes, ALP or alkaline phosphatase. So whenever there is disease of leukocytes, say about you can have the say about CML, chronic myeloid leukemia, the cancerous condition of the leukocytes. The concentration of ALP decreases. If the person is having lymphomas, okay, then the concentration of this type of isoenzyme, leukocyte derived ALP, will be elevated. So these are the 6 different types of ALP. What are they? Alpha ALP, that is also called as alpha 1, alpha 2 heat labile, alpha 2 heat stable. Next, pre-beta and gamma ALP. Last one is leukocyte derived ALP. So these are the 6 different types of isoenzyme. And there is also one more type called as Reagan isoenzyme. In some particular cancerous condition, okay, in particular cancerous condition, like say about cancer of lung or liver or GI tract, there is elevation in a type of called as placental type of ALP. When it is elevated, when there is cancer of this tissue, when there is cancer of this tissue, there is elevation in type of isoenzyme which is called as placental form. And this kind of isoenzyme we call as Reagan's isoenzyme. Normally, it is not seen. In adults, you don't see placental type of ALP. In adults, if you see placental type of isoenzyme, which we call as Reagan's isoenzyme, it should dictate you that there is some cancer which is going inside the body. Specifically, it can be from the lung tissue, liver tissue, or gut, GI tract. So this is called as Reagan's isoenzyme, or also called as carcinoplacental isoenzyme. Okay, what is called as carcinoplacental isoenzyme. So these are the different types of isoenzymes of ALP, and also a special note on Reagan's isoenzyme. So if I tell you again how they are differentiated ALP is based on electrophoretic pattern, whichever runs near to the positive node, we call alpha, like proteins we have seen, alpha, beta, and gamma portion. So alpha, you can have alpha 1 ALP, alpha 2, you have 2 different types, alpha 2 heat stable and alpha 2 heat labile. Say this is beta, but this isoenzyme is just before the beta, pre-beta. Okay, pre-beta isoenzyme, and there is gamma isoenzyme. Okay, gamma ALP. And other last type is LAP, leukocyte derived alkaline phosphatase. So this is about the alkaline phosphatase, which is a major exam question. And also note on Reagan isoenzyme is a 3-mark question. It is also called as the carcinoplacental isoenzyme. Between all these things, this is elevated only in cancer. Okay, in cancer, you identify type of ALP is Reagan's isoenzyme. Others, it can be disease or disorder of particular tissue. Okay, all 6 isoenzymes. So that is about the ALP, alkaline phosphatase. Now, last one, alcohol dehydrogenase. Okay, alcohol dehydrogenase. What is it? It is a heterodimer, means it contains 2 subunits. Okay, there are 2 types of lactate alcohol dehydrogenase. Okay, what are they? Either they are alpha beta 1 or alpha beta 2. Alpha beta 1 is slow metabolizer. Okay, slow metabolizer. What is this helping in reaction is it is converting alcohol into acetal acetaldehyde. That is alcohol dehydrogenase. Okay, ADH. Sorry. Alcohol dehydrogenase. Two isoenzymes, 1 and 2. Alpha beta 1, alpha beta 2. Alpha beta 1 is slow metabolizer. Alpha beta 2 is fast metabolizer. Slow metabolizer is seen in white Americans. Okay, white Americans and Europeans. The Western countries predominantly have first type of ADH, alcohol dehydrogenase, that is alpha beta 1. The Asians, Japanese, Chinese, say about Chinese or South Asians, all have the alpha beta 2 type, second type of ADH, that is alcohol dehydrogenase, which metabolizes the alcohol very fast, which increases the concentration of acetaldehyde, which is not non-toxic. It is a toxic. So that's why when you compare the Westerns with the Easterns, if the same concentration of alcohol is taken, the toxic effect, the short term or the long term, is majorly seen among these areas of the countries, because we predominantly have this type of isoenzyme ADH2, which metabolizes the alcohol to acetaldehyde very fast, and acetaldehyde is a very toxic compound, which will have its signs like, for example, increase in heart rate, tachycardia, or also like say about flushing. All these symptoms are predominantly seen with same amount of alcohol very fast in these part of the countries when compared to the Western countries, because they contain an enzyme which metabolizes very slow. Alcohol is metabolized very slow, whereas the alpha beta 2, that is ADH2, is seen in the our countries. That is fast metabolizer, because of which the concentration of acetaldehyde elevates, and it will cause the signs and symptoms which are very toxic to the body. So that is about the isoenzymes. So this we conclude with the topic isoenzymes. So to summarize, isoenzymes are the the same enzyme or multiple form of enzyme which takes part in same type of reaction. They are physically different, chemically different, then electrophoretically different, chromatographically different, immunologically different. Apart from this, they are also differing in Km value, that is Michaelis constant value. They are differing in the maximum velocity what they can achieve, susceptibility towards inhibitors, and also they differ in the degree of denaturation. And they are also specifically to tell they are synthesized from different tissue or different cells. In the cell, they are synthesized or coded by different genes. So that is about isoenzymes. Examples for the isoenzyme, majorly lactate dehydrogenase, alkaline phosphatase, creatine kinase, and alcohol dehydrogenase. Alcohol dehydrogenase. LDH has got 5 different types of isoenzymes. ALP has got 6 different types of isoenzymes. CK has got 3 isoenzymes. Yes. And alcohol dehydrogenase has got 2 isoenzymes. Yes. How many subunits do LDH has? Which is HH2, all 4 M subunits. Yes. So 5 isoenzymes from 4 subunits. ALP, it is only 1. It differs only in percentage of carbohydrate which is present in the structure. CK has got how many subunits? 2 subunits. Either they are made up of M or B. It's a combination of M and B. ADH, it contains alpha and beta. They're either alpha beta 1 or alpha beta 2. So this is about the isoenzymes. And also you should know their significance. Their significance. Like we have discussed in myocardial infarction, what is the pattern of LDH, what is the pattern of CK, creatine kinase, and alcohol consumption, ADH, then alkaline phosphate in various like obstructive jaundice, what type of isoenzyme is elevated. If it is, say about the liver, leukocyte cancers, lymphoma or CML, what is the pattern, and what is Reagan's isoenzyme. So that all are the subparts of the alkaline phosphatase. So is it clear with the isoenzymes for today? Yes. So lastly, what you can expect, as I already told in the university questions, long essay, they can ask you define isoenzymes. So you should write what is an isoenzyme and characteristics, and describe different types of isoenzymes. So when long essay like 15-mark question is there, you should write at least 3. What are LDH, CK, and ALP, isoenzymes of all these 3. Or sometimes in specifically for 5 mark, they might ask you write a note on isoenzymes of LDH. So you should describe what is LDH, what type of reaction takes place, then what are the subunits, what is the origin of subunits, and how they are different in tissues, and what is the significance, like what disease you can identify by using these subunits. Same way, write a note on isoenzyme creatine kinase, or also called as CPK, creatine phosphokinase. So again, you should write what type of reaction it is involved in, how many subunits are there, what are the different 3 isoenzymes, which tissue is originating from, and what diseases it can help in the diagnosis of the disease or disorder of that tissue. And last, note on Reagan's isoenzyme, which is nothing but related to alkaline phosphatase. Okay, alkaline phosphatase. Majorly it is seen in cancerous condition. Okay, that is cancerous condition. Elevation in. Secondly, it is seen in adults. Most commonly in adults, you see placental origin of isoenzyme. Normally in adult, there is no placenta. Definitely this kind of ALP should not come into the tissue or in the blood. If you see placental origin of ALP, then it is nothing but the Reagan's isoenzyme, which is the carcinoplacental type of isoenzyme. Okay, so this is about the isoenzymes for today. Fine. Is it clear for